maybe later this year. All right, well, shall we begin? It's 12:01 here in Okinawa. Sound good? Is recording
Sounds
set
good.
up?
Yep,
Perfect.
we're good to go.
Awesome. Well, thank you, Courtney, and thank you all for joining us today on the inaugural session of this new population biology symposium or seminar series. I guess you could call it like a remote symposium that is, never ending, perhaps. But I'd like to thank Courtney for kind of spearheading this initiative. It was really just all her idea, and I remember, after our session at the Ecological Society of Australia meeting, you mentioned, how it would be good if we could run a seminar series in a similar vein to some of those that are being run on the opposite side of the earth, but that we really have a difficult time accessing because of, time zone limitations and everything. And also just given the fact that across South Asia, East Asia, Oceania, it's quite difficult. You can't just, hop on a bullet train and ride to Brisbane or Calcutta, for instance. So remote seminars of this vein, I think, are much more important to be done in places where travel is limited and funding is limited. So yeah, we're going to be doing this once a month. It will primarily be on the last Thursday of each month. So today is sort of an exception. And if you haven't already signed up, please sign up for the updates. And there's an email listserv that has gone out. And maybe we can post that in chat as well if you haven't already signed up for that. And so with that, I'd like to welcome Ryan Chisholm, who will be our first speaker in this series. And so I've known Ryan's work for a long time. We've never actually met in person, though. But yeah, I've followed his work on tree demography and really doing a lot of great work linking ecological theory to some of these large-scale data sets, like tree inventory data sets on Barrow, Colorado Island, even just like, global tree inventory stuff. Really, really cool work linking data and theory together. He's made a lot of big contributions in niche theory and neutral theory on both the theoretical and empirical sides. He's currently zooming in to us here from Singapore, from the National University of Singapore, where he's an associate professor in the Department of Biological Sciences. And he's previously worked at institutes such as Princeton University and the Smithsonian Tropical Research Institute on Barrow, Colorado Island. Were you living there at the time?
As a theoretician, I was not. I was living in the city, but I would go there once a week for a seminar. Yeah.
Great. Which I encourage you all to visit if you ever get the chance in Panama. So today he'll be presenting a unified theory of niche assembly and dispersal assembly in community ecology. Very interesting stuff. And I just want to remind you all that the talk will be recorded, but the question and answer session will not be. So feel free to, ask away. Great. Take it away, Ryan. Thank you.
Thanks, David. Yeah, and thanks everyone for coming virtually. And I only realized this morning when I was rereading my emails that this is the first seminar in the series. So I won't feel any pressure, but I'm very honored to have been invited. And in fact, this talk is really a modified version of a talk I gave at a conference in Boston a couple of years ago on predictive ecology. And I think there are 100 and something attendees, and there were only two of us from Asia, me and a colleague from India. And I commented at the time in my talk about how a nice thing about the northeastern US is you have this concentration of big institutions all within driving distance. And other people I was talking to, they'd be like, oh yeah, so it's in Boston. People say, oh, I drove from Montreal. Oh, I drove from Baltimore or whatever. And there's this kind of critical mass of people studying topics relevant to quantitative ecology that I think is quite stimulating. And having did my PhD over there, and so I can really relate to that. And I said one thing I feel over here is it's a bit more isolating, which is not necessarily a bad thing because it's not too many distractions, but sometimes I think we should make more of an effort to connect with people in the region, even though it's a bit harder. So I think this is a great step in that direction. And I don't know what your guys' long-term vision is, but maybe even if we could try to do like at least a once every couple of years in-person meeting or something, try and get some more collaborations going. As you can probably tell, I'm from Australia, so I'm always keen to strike out more collaborations back home. But with that aside, let me get into the meat of the talk. So this is the title, A Unified Theory of Niche Assembly and Dispersal Assembly and Community Ecology. And I'll be describing work which goes actually all the way back to my PhD, but mostly focused on more recent stuff I've been doing. Okay. All right, so the big question I'm going to be focused on for the talk is what maintains the diversity of local ecological communities. And there's really two broad possibilities. The first is that it's niches, so local processes, and the second is that it's dispersal, so organisms coming in from the outside, which then ultimately puts the explanation onto regional processes. And these are not mutually exclusive. It may be a combination of both or more one than the other in different contexts, but that's what we want to figure out. Just to give you a classic example of niche theory, this is one of my favorite niche theory papers, Dave Tillman's classic paper from 1981, where he had a resource competition theory, and he used that to predict the outcome of competition between pairs of algal species. And he parameterized the model based just on single species experiments. And then he was able to make a prediction for what the outcome of competition would be for different combinations of these two resources, so silicate and phosphate. So you can see here in this wedge-shaped region, he predicted coexistence, and in the other regions, he predicted different outcomes. And that was purely, as I said, that was purely, this prediction was purely made based on the single species experiments, but then he actually ran the two species experiments, and these graphs show the theoretical predictions as curves and the actual experimental data as time series, and the three different scenarios for three different combinations of resource concentrations. And you know, for these being true predictions, as opposed to just tuned fits of models to data, I think this is pretty good. Right? So this is a long time ago now, but I still think this is one of the best niche theory papers that I've ever seen. And one thing I always wonder is, well, you go back in time to 1981, and you might think by 2025 we would have this all figured out by now. We'd have extended this to multiple species. We'd be able to predict the dynamics of whole communities of species, but we still can't do it. So maybe that's a question for discussion time. Why has this not progressed as far in this direction as one might have anticipated back in 1981? But I think, just briefly, the problems are, the curse of dimensionality, and these systems become complex, noise becomes important, it gets much harder to predict them once you have multiple species. And then a bit more recently, Hubble came along with his unified neutral theory of biodiversity and biogeography, and he upset a lot of people by throwing all that niche stuff out the window and just saying, well, what if we assume that all species are equal on a per capita basis, and we focus instead on processes like dispersal, stochasticity, speciation? What kinds of predictions can we get from a simple neutral model like that? And I'm not going to go much into the details of neutral theory. I did publish a recent book chapter in, there's a volume to celebrate 100 years of research on Barrow, Colorado Island in Panama. I wrote a book chapter about neutral theory and BCI there, if anyone wants to look at it. But I'm just going to highlight one of the signature achievements of neutral theory, which was to accurately predict the species abundance distribution of trees on Barrow, Colorado Island. So this is for a 50-hectare plot in Barrow, Colorado Island, where they go and measure and census all the trees every 5 years. So this is this is abundance here, number of individuals on a log scale, fraction of species. The gray bars are the data, and the neutral model gives a very good fit to this. And in fact, in this case, with no fitted parameters. The original model had 2 fitted parameters, but work done by myself and others managed to estimate those parameters from independent data. And so you get this this true prediction here, and it looks pretty good. Now, I should say neutral theory fails in other ways. So it has some predictions that work very well, some that fail spectacularly. But I think that's a strength rather than a weakness. It's an actual predictive theory that you can confront with data. And where you see discrepancies with data, that means you're missing some process and you need to go back and modify your model. So I think even though neutral theory itself has limitations, it represents a rigorous approach to science. Okay, so back to our broad possibilities. What maintains diversity? Niches, right? So to understand niches, we use niche assembly theory, or it could be dispersal. And in that case, we draw on dispersal assembly theory, which is not just neutral theory, but mass effects, source sink dynamics, other terms you've probably heard. Right, so I should just going back to here, the process which is maintaining all these species in this in the system in this model is dispersal from the outside. So I said you have this 50-hectare forest plot, and as opposed to a niche model, in a niche model, these species would be all maintained by stabilizing niche mechanisms. In a neutral model, all those species are maintained from dispersal from beyond the plot. That's why I classify it as a dispersal assembly theory. Okay, so I've been working on both branches of this theory for a long time. And an early paper I produced, which is very theoretical, it was in the Journal of Theoretical Ecology, the Journal of Theoretical Ecology. And there's no data here, but we're just looking at a hybrid niche neutral model. And let me take a second to explain this. So this model has a parameter, which is the speciation rate here, per capita speciation rate. So it's very small. And here you have species richness in the model. And the solid line shows the neutral prediction. So in the neutral prediction, as the speciation rate becomes very small, you just collapse to one species, right? A monodominant system. But in this hybrid model we built, where you have some niches, and then you have neutral dynamics operating within each niche, when the speciation rate is very high, the niches don't have much effect, right? Because the niches are overflowing. You have more than one species per niche. But as you bring the speciation rate down, whereas in the neutral model, the diversity would collapse to 1, in the hybrid model, it doesn't, right? Instead, it asymptotes to the number of niches. And it's quite clear why that happens. I mean, this is kind of built into the model. You set the number of niches, I think it's 16 in this case. And when you have low diversity, that provides a lower bound on the number of species. So this paper was more illustrating these concepts rather than trying to make any big predictions about really ecological systems. And in fact, when I wrote this paper, I never thought we would actually test this against real data. But then sometime later, I was browsing through MacArthur and Wilson's classic book on island biogeography, and I saw this graph. So this is an island species area curve, which probably many of you have seen this kind of thing before. Number of species, log acres. This is for plants on islands in the Kapinga-Marangi Atoll in the Pacific. And what you see here, I mean, this is the original graph. You have this increasing species area relationship here, which is typical. That's what you'd expect from island biogeography. But for small areas, there appears to be no relationship between species richness and area. And this has been termed the small island effect. So it's been known for a long time. People had various explanations for it, but I never really bought those explanations. But when I saw this, browsing through MacArthur and Wilson, I had this flash, and I thought, I know what's causing that, right? And it's related to that previous theoretical paper, but it's a bit different, because on that previous theoretical paper, the variable I had on the horizontal axis was the speciation rate. Here we've got area. But I'm going to explain in a bit how these are related. But first, I want to introduce a version of the model that we use to explain this. Okay, so this is a cartoon model, but it's underlain by rigorous mathematics. I've got this island here, and I'm just stipulating that there are 4 niches on the island here, indicated by soil types, although there could be other kinds of niches. And the rules are going to be the same as the rules for that other model I mentioned. You have a set of species in each niche, and each species can only live in one niche, and the niches can't interact. but you can have more than one species per niche. I'm just saying arbitrarily the area is 100 units, whatever units we're working in. Got 4 niches for the 4 soil types. And you will always, in this model, you'll always have at least 4 species, 1 for each niche, as I've illustrated here. And then as the dynamics of the model proceed, you get birth and death events. So within each niche is neutral. And as you go to a larger island, you still have those same 4 niches. You get death events, and occasionally those death events will be replaced by immigrants. But when your island is fairly small and isolated, those immigration events are so few and far between that the drift process, or I guess in a more general model, the competitive exclusion process, drives all but 1 species to extinction within each niche. So most of the time, you just have 1 species in each niche. Occasionally, you get an extra one coming in through immigration, but those events are so rare that at the dynamic equilibrium, they'll usually just do 1 species per niche. Okay, so we're back to 4 species again there. As you go to even larger islands, and this is a bit of a sop to the niche people, I say, well, yeah, sometimes you'll get an extra niche, but I don't think, so I'll put an extra niche there, but I don't think that's under the theory anyway, that's not a major factor driving the increase of species richness with island area. And so if we just construct a species area curve for the cartoons I've shown so far, and some extra points I didn't illustrate with cartoons as well, we're just stuck at around 4 or 5 species on each island, and there's no increasing effective area yet. Now I've gone to a bigger island, and so we still have these occasional immigration events, but now the island's big enough that firstly, it will take longer for the extinction process to play out within each niche, and secondly, because the island's bigger, it's a bigger target for immigrants, that we may end up with more than 1 species per niche on average at the dynamic equilibrium. So here we've still got 2 species in the sandy niche. The extinction hasn't occurred yet for one of the species, and now I've got a 3rd species immigrating here. Right? So once you reach this regime, you're no longer stuck at that lower bound, and you actually get a non-trivial colonization extinction dynamics within each niche. Right? And that same thing in that other niche. And as you go to even bigger islands, this process just becomes more and more important. You get more immigrants, the extinction process takes even longer, and so you just get more and more species as your island area increases, consistent with traditional island biogeography theory. Okay, so if we draw the species area curve, once you enter this regime, you're in the traditional island biogeography regime of increasing species richness with area. And as I said, that was a cartoon, but we had some rigorous mathematical model behind it. This is the formula with these digamma functions. I don't expect you to parse all of this in the short time I'm showing this slide, but I just want to highlight that there are only 3 free parameters here. The n star, which is the number of niches, m, which is the per capita immigration rate, typically be pretty small, and theta, which governs the meta-community diversity. And so my insight was that this model could explain this pattern. And just going back to that earlier model with speciation rate, here, what's mediating this transition? I mean, it's area, but the mechanism we're assuming is immigration. So as these islands get bigger, the immigration rate, the total immigration rate increases, and that's what causes this transition. Okay, from a niche assembly phase down here, where you have low immigration, to a dispersal assembly phase up here, where you have high immigration. And so we went and fit that model to 100 different archipelago data sets. This is just 4 of them. And you can see it provided a pretty good fit. Right, so the R squared we got for these data sets was higher than people had got before for the same data sets. And plus, this was a mechanistic model. So in the past, people had tried to fit breakpoint regressions or polynomials to these data, and just phenomenological models, and actually got a worse fit than we got. All right, so I thought I was pretty pleased with that. I thought that was published in Proceedings of the Royal Society about a decade ago now. But I presented that work in many places, and so I've thought a lot about the strengths and weaknesses of that. the strengths, as I was saying, you have this mechanistic model that predicts the functional form, right? You're not relying on just some phenomenological model with no basis in the mechanisms. Some weaknesses, I mean, we do have 3 fitted parameters, and as we all know, if you have enough parameters, you can tune the model to lots of different patterns. And also that this is a hindcast rather than a forecast, so where, I talk a lot about prediction and ecology. I mean, this is maybe prediction in inverted commas, but the data's already there. Right, so it's not quite as impressive as if you predicted something and then went and got the data to test the prediction. And this one is one that comes up a lot, causality. So as I mentioned, we're assuming that the mechanism which is driving this change in the species area curve here is immigration, and that's mediated by area. So we're assuming that area is a proxy for immigration, but area is confounded with all kinds of other things. So people would say to me when I present as well, you're assuming that niche diversity doesn't increase much with area, but maybe it does. Maybe this is all driven by niche diversity. And I could rebut those points verbally, but I do take the more general point that we're using area as a proxy here, and it would be nice to look at immigration directly. And so in a theoretical paper published a bit more recently, we went back and thought about this. If you just had, if immigration rate was the thing that you were able to control under pure niche assembly theory, you wouldn't necessarily expect any relationship of species richness to immigration rate, because the species richness is determined by the niches under niche assembly theory by definition. Under dispersal assembly theory, you'd expect some sort of monotonically increasing relationship of species richness to immigration rate. And the question is, how do you combine those? Now, under classic theory, so if you look back at some of the old MacArthur and Wilson papers where they try to reconcile niche theory with the island biogeography model, the assumption is that this is the classic island biogeography regime. When you're on isolated islands, your niches are not full, and so your number of species is just determined by how much immigration you have. But if you have an island that is not too isolated or is very large, then theoretically they would say you'd be in, then your niches would be full and you'd be in the niche assembly regime. So this is the classic prediction, and there's actually mathematical models in these papers that predict a shape like this. But based on our novel theory, so this is a paper with my postdoc TAC form in 2021, and this is just based on what I've been talking about today, but instead of having area here, we're putting immigration rate directly. So under our theory, we're saying when the immigration rate is very small, you just be stuck with the minimum number of species, which is however many niches you have. But then as you increase your immigration rate, those niches will start to overflow and you enter this dispersal assembly regime. Again, analogous to that very first result I showed you from my early paper where we had speciation rate here, but now it's there we were thinking on a very large scale in terms of speciation. Here we're thinking of on a small scale, so we're looking at immigration. And this is where Lynette Lope comes into the story. Maybe some of you know her. I think she's been involved in setting up this talk series, but she's teaching right now apparently. And so Lynette came to me a few years ago. We'd collaborated on a couple of previous projects, and she was looking for some new idea, a new theoretical prediction to test. And I'd thought maybe we'd do this with microbes or something one day if I found the right collaborator, because microbial systems are fairly easy to manipulate. But Lynette was convinced she could do this with her intertidal system. And I have to admit, I was a little bit apprehensive because I don't do experiments. They seem very tricky and complicated, but she was determined. And so we talked a bit about experimental design and how many replicates we'd need and so on. And I think her main innovative idea was to use these tiles that she'd been using for other experiments, but to put boxes over them with holes in the polycarbonate panels. And by manipulating the number of holes, she could control the immigration rate. So we're using holes here as a proxy for immigration. And I should say these units are underwater 95% of the time. So the photos show them out of the water because that's easier to photograph, but most of the time they're under the water. So you have creatures moving in and out through these holes. So each treatment replicate is a habitat tile with its stainless steel cage and the polycarbonate panels. And she can vary the number of holes from 1 to 45. Okay. These are some of the creatures that she observed. And she went out and ran this experiment over the space of a year. And she can correct me on this. Well, she's not here, but I think she's going to watch it later. I think she was out there nearly every week, maintaining the units and counting the organisms inside the units. I skipped over this bit. So there were 12 treatments with different numbers of holes ranging from 1 to 45 and 5 replicates for each treatment level. And just to remind you, these are the 2 alternative theories. So we had this classic unified theory where this relationship of species richness to number of holes or immigration rate should be saturating. And then the novel theory where it should be flat at first and then increasing without any apparent bound. And these are the results. These are showing the same data on both panels, but with fitting the classic model here and the novel model here. And what I like about this, I mean, yes, the novel model fits better, but even from eyeballing it, you can just see what's going on. I mean, here you go from 1 to 5 holes. So you're increasing the immigration rate by a factor of 5, or at least the proxy for immigration by a factor of 5, and there's barely any change in species richness. And the classic model is completely unable to capture that, whereas the novel model captures it quite nicely. And at the other end, the classic model predicts that species richness is going to saturate this value of about 16 here. But actually, when we look at her data, after 12 months, the species richness has stabilized in most of these treatments, but it was actually still going up in these treatments. So I think if you run it for longer, the fit of the classic model would look even worse. Okay, so we have evidence of saturation at a low number of species at this end, but no evidence of saturation at this upper end. So it was quite consistent with our novel model that was published in Nature about a year and a half ago now. Okay, so it supports the novel model. So again, stepping back and thinking of some of the strengths and weaknesses of this. Strength is this is a true prediction. And I should say this is really exciting for me being involved in this collaboration, because as a theoretician, I'm usually stuck either working with no data or working with existing data. And to have this project where we're like, okay, this is the prediction, what experiment do we need? Let's go and do it, see what we get, was seemed very risky and exciting. Okay, we're predicting the functional form as before. It's a risky experiment. It could have been substantially wrong. I sort of said to them, well, in theory, this should work. I'm pretty sure that this theory is right, but we could have easily got something completely different. But in the end, it worked. Weaknesses: we still have these fitted parameters. Okay, maybe in the future we can do something about that, trying to estimate those parameters from independent data. And also, we're predicting just this one pattern, the species-area relationship. Lynette's now planning to do more work actually in Victoria. She's a lecturer at the University of Melbourne now. She's planning to do more work spreading units over a broader area and looking at turnover patterns in addition to the species area curve. Okay, so the last bit I want to talk about, how am I going for time? All right, going all right. I've been talking about species area relationships for islands, but more typically, when we're talking about species area relationship, we're looking at mainlands, looking at nested areas within mainlands. And the classic shape of the species area relationship on a mainland is this triphasic curve. This is for birds starting from a small area in, I think that's Ohio, and this one's a small area in Panama, up to the scale of the entire world. So this is log area here. These graphs were produced by Preston in 1960. And you can see this very steep phase here that we call this the sampling phase. You see another very steep phase at large areas. That's when you're at such a large scale that an increase in log area reveals completely new biogeographic realms, so lots of species, lots of new species. And then you have this power law phase, this flatter power law phase that joins the two. And the slope here will be, the exponent of that power law will usually be in the range of like 0.2 to 0.3. And many of you have probably worked with the power law or species area relationship before. But how do we reconcile this with these island species area relationships? Is there sort of some unified theory of species area relationships? Okay, so we looked at this in the same theoretical paper I was talking about before. And we looked at a large-scale version of this model, and we predicted actually a 4-phase, or what do I call it, a tetraphasic species-area relationship. So you have these classic 3 phases: the sampling phase, dispersal assembly phase, and the speciation assembly phase. That's how we refer to the very large one. And in theory, there's this niche assembly phase here, which corresponds to that small island effect we've been seeing, and also that flat phase we saw in Leonard's experiment. But according to the theory, most of the time you don't see this because in a mainland context, immigration is very high. So if you think about Leonard's experiment, we artificially restricted immigration. If you think about the small islands, they just naturally have low immigration. But when you're in a mainland context, like a 50-hectare forest plot in Panama, that community is constantly being bombarded with lots of propagules from the outside. And that's enough, under our theory, to completely hide this phase. And so then what we said was, well, when should we be able to see this phase? And in theory, you should be able to see it if you have a system with low meta-community diversity. Another option, which I haven't written here, is a system with very low dispersal. Right, but the one I'm going to look at is low meta-community diversity, because if you have low meta-community diversity, then, okay, so you have a lot of immigrants maybe still in terms of propagules, but they're all the same species immigrating over and over again. And so in that case, in theory, you should observe this niche structured phase. And the system we chose to look at this was mangroves. Right? So mangroves, they grow in this intertidal environment, and they have notoriously low global diversity. So there's only 70-something species worldwide. And so they were a suitable system for looking at this. And we kind of know a priori, just from personal experience, that most mangrove communities do exhibit quite low diversity. Right? But the question was, does the species area curve saturate at some low value that is not 1? Right? If it saturates at 1, we can just trivially reconcile that with standard theory. But what we're looking for is a niche structured phase where it asymptotes some low value that is not 1. Okay, so we had this mangrove data set. This is from collaborator Andre Rovai, who is now at the Smithsonian in the US, and Dan Fries, who is at Tulane University in Illinois. And the initial part of this work was actually done by my honor student, Nicholas Fong. Okay, so this is the result. We constructed this species area curve from a very small scale here. So this is 10 to the minus 4 square kilometers. What's that, 100 square meters? So that would be 10 meters by 10 meters, all the way up to the entire world. This is for the Indo-Western Pacific, but we have a similar graph for the Atlantic, Eastern Pacific. And what you see, I mean, yes, the species area curve is increasing, but if you look at this over 10 orders of magnitude here, it increases from maybe about 3 species to around 4. That's a negligible increase. According to the classic power law species area curve, over 10 orders of magnitude, you'd expect species richness to increase by about a factor of 300. Right, so this is a glaring exception to those power law species area curves. And importantly, as I was saying before, this doesn't just asymptote to 1 as area becomes small. It's stuck at some value greater than 1, roughly 2, 3, maybe 4. And our interpretation of that in the context of the theory is that there's about, say, between 2 and 4 niches for mangroves in the typical coastal environment. And that's actually consistent with the standard biological understanding of these systems, that you have these different mangroves are adapted to different tidal ranges. And so in the context of our theory, we'd say, even if you have very, very low immigration, you're always going to get sort of 2 to 4 mangrove species because they split up the environment by tidal zone. But rarely do you get more than that because immigration of different mangrove species is just too uncommon. But in principle, if you did have enough immigration, you could overwhelm those niches and you get, there's still the same 2 to 4 mangrove niches, but in theory, if you had more immigration, they would overflow and you could get more species per niche. Okay, so we've revealed this hidden niche structured phase of the species area relationship. Okay, so implications of all of this. We have this novel unified theory that predicts that communities are niche assembled under very low immigration and dispersal assembled otherwise. And all of these studies that I presented, they suggest that niche diversity is usually low. And if you think back to the small island effect, it usually saturates at between 5 and 10 species. In Lynette's experiment, her niche restriction phase had about 5 species. In the mangroves, it looks like maybe between 2 and 4 species. So going back to the very first thing I talked about at the beginning with Tillman's experiments, and he was looking at how pairs of algal species could coexist, I think under niche theory, it's very easy to show how a handful of species will coexist by resource segregation or some other niche mechanism. But if you play with those models, it's actually hard to get lots of species to coexist. So I think niche theory can get us to about 10 species within a trophic level a lot of the time, but it's hard to get beyond that. To get beyond that, we really need to invoke dispersal theory. And then if you think about most local communities on mainlands, well, if we extrapolate what we're seeing in these systems that we've been studying to general local communities on mainlands, we'd say they have just a few niches, 10, maybe fewer. And the reason they have so many species, that 50-hectare plot in Panama has about 300 tree species. The reason it has 300 is mainly because of dispersal assembly. It's constant influx of propagules from the outside. And I think that's why neutral theory does so well in many cases in predicting various patterns for those high diversity communities. Okay, that's my lab. I want to thank my lab and my funding sources. And last, a bit of a plug for my textbook, which is coming out later this year with Princeton University Press, Theoretical Ecology, Concepts and Models with R. I'm just going through the proofs right now, which is a bit of a painful exercise with all the equations and the code, but I think it's going to be worth it. So I guess we can